Thank you, Debbie. So yes, like she said, my name is Conor Howard. I'm a scientist at a small molecule drug discovery company in California called Octant. Today I'm going to be talking about some deep mutational scanning work we did in collaboration with Bristol Myers Squibb on a protein called TYK2. But first I want to take a step back and talk a bit about autoimmune disease. So autoimmune diseases are a pretty complex set of disorders that range in severity, clinical outcome, and phenotype, and their prevalence is on the rise. So there's a good chance that you or someone you know is suffering from a disorder that falls into this broad class of diseases. Fortunately, in recent years, there's been a surge in therapies that are hitting the market to treat autoimmune disease, but they come with pretty severe side effects. The first obvious one is that when you try and target the immune system or downregulate it to dampen autoimmune disorders, you increase the risk of infection, severe and oftentimes lethal infection. But in addition to that, you also carry a high risk of blood clotting, heart disorders, and cancer. And so for these reasons, the FDA slaps pretty severe safety warnings on these treatments, and the barrier to entry for patients is pretty high. That's where recently TYK2 has come onto the scene as as an exciting, potentially safer therapeutic target for modulating the immune system. So TYK2 is a Janus kinase involved in cytokine signaling. It's essential for immune function, but it's not, downregulation or modulation of TYK2 activity is not associated with these side effects I just mentioned, even though all the other proteins in its JAK family are. And even more excitingly, there are human alleles in the population that have a protective effect against autoimmune diseases. So that means there are people out there who have a minor mutation in TYK2 that never get autoimmune diseases and also never have an increased risk of infection. So for that reason, it's really, really important for us to try and understand both mechanistically and also at a genotype-phenotype level, what is the relationship to TYK2 and the immune system in general. One thing I'll say really quickly right now is that a huge branch of this project, or a big part of the motivation, was to establish exactly that, to build and define a genotype-phenotype DRC so that we can map all of the possible variants and see how they relate to clinical outcomes. I'm not going to talk about that today. I'll say that for another time, but feel free to come find me after the talk. Instead, I'm going to talk about some more drug-specific experiments we did. So like I said, we're a drug discovery company, and we love multiplex technology in general. We really like trying to push the boundaries of using it for drug discovery-specific purposes. What do I mean by that? Today, I want to touch on two things. One, this idea of allosteric site identification. Can we use multiple different DMS methods to uncover sites that are potential therapeutic sites of actions, protein-protein contacts, catalytic and allosteric sites, things like that, starting to disentangle the mechanisms of gain and loss of functions. And then two, at Octant, we do a lot of high throughput chemistry, and what that usually entails is taking a protein and scanning thousands to hundreds of thousands of slightly different compounds. But we're really interested in seeing if we can do the inverse of that. Can we take our favorite 3 compounds in-house for our current program and just do a ton of DMSs? And learn from the results how these ligands and drugs interact with these proteins to better understand how to optimize them in the future. So to this end, for this project, we employed 2 specific DMS assays. One is a signaling assay that interrogates IFN alpha signaling, and the other is a general protein abundance assay, very VAMP-seq style, where we just flow GFP tag TYK2 to measure abundance. For IFN alpha is a cytokine. It is released upon infection, binds cytokine receptors, triggers cascades that are meant to respond to infection, but also increase inflammation. So we developed a transcriptional reporter that reads out on the activity of this pathway, of which TYK2 is crucial. We do all of this work in our favorite landing pad cell lines, where we can integrate single copy variants linked to a very specific unique barcode. And because TYK2 is a pretty big protein, our libraries ended up being massive. So we have over 20,000 protein variants, over 50,000 codon variants, and about 99.8% or better coverage across the libraries. We also use multiple barcodes per variant. So what I mean by that is each variant has around, if not more, 30 barcodes, unique barcodes per variant. those all are integrated into separate cells and can be grouped or treated as separate pseudoreplicates later on. One thing I'll also say right here is that our data science team has done a lot of really good work to develop some models, specifically mixed effects models, that really, in combination with our coverage and depth, give us a lot of statistical confidence and power in the kind of effects we're seeing. As an example, we're able to call 10 versus 40 or versus 60% loss of functions, where that is really important when you're looking at very subtle effects, whether it's drugs or genotypes and phenotypes. All in all, we tested 16 unique experimental conditions, and over 10 million data points were collected over the course of the 2 years. So just jumping right into the data, here I'm showing you 2 heat maps from some of the experiments. The one on top is for the IFN-alpha signaling assay. The one on the bottom is the protein abundance assay. Secondary structure of TYK2 is overlaid on top. Immediately, there's lots of fun patterns and things that you can jump at and think about. One of the obvious ones is that the loss of functions, which are shown in blue here, tend to be enriched in structural domains. That makes sense, and especially in the pseudokinase and kinase domains. But what was more interesting to us is looking at where these maps differed, where they disagreed. Specifically, where there were effects that only hit the signaling assay and not the abundance or stability assay. So here I've highlighted 2 clusters of positions that seem to have loss of functions for the IFN-alpha assay and no effect in the abundance assay. This is really cool and exciting because, as a few other speakers have mentioned, it's starting to give us insight into the mechanisms of the loss of function. And in these cases, we can say, okay, whether it is some expression regulation or an actual stability effect, that's not happening here. And these sites are likely involved or responsible for things that are catalytic, allosteric, or protein-protein contacts. We can do this comprehensively and systematically across the whole protein and start to evaluate where they lie in space. So here is a cryo-EM model of just the kinase and pseudokinase domains of TYK2, where I'm showing positions colored in blue as positions that are enriched for these signaling-only loss of function effects. And one neat thing that jumped out immediately is we captured 2 sites that we expected to see. Namely, on the left, the orthosteric active site, the site that actually does the business work of phosphorylating substrates, but then on the right, the allosteric inhibitor site. I call it the allosteric inhibitor site because some of the most recent inhibitors developed against TYK2 have been found to bind in this region of the pseudokinase domain and actually act to inhibit the protein by more tightly binding the kinase domain and preventing it from acting. But I want to emphasize here, we did this assay in the absence of drug. And so what's really cool is that each one of these variants on its own is able to recapitulate the molecular changes or mechanisms that are happening when these inhibitors bind the same site and are transducing that inhibitory effect through the 2 domains. And then in addition to those known sites that we expected to see, we also captured some unknown regions, both of them at the periphery, but also in the contact regions of the 2 domains. And in particular, there are 2 that basically every single variant falls into this signaling only loss of function category. I don't have time to get into it, but we also extended this analysis to the entire full-length protein, and we're able to recreate protein-protein contacts with the cytokine tails, STAT proteins, and other things. So this has been really exciting to kind of just have a top-down view of, okay, where are the sites of action? What are the things that we know? What are the things that we don't know that we could use to target new therapies for? Could this have expedited the 20-year process of developing the therapies that are on the market today? So next I want to jump into this idea of protein SAR, or DMS screening of protein-drug interactions. So up until now, I didn't show you any data with drugs. We decided to put some drugs into the mix and say, first, okay, can we add one of these inhibitors that binds that site and find drug-resistant variants? By that, I mean, at very, very high concentrations, are there any variants that can prevent this drug from actually working? And indeed, we do. So on the left here is a volcano plot where highlighted in red are the very small number of significant variants that have activity even in the presence of inhibitor. And they all light up smack dab at the depth of the binding pocket. So this was really exciting result to see. Actually, my data science partner on the team didn't know the structure of the protein, didn't know anything about the experiment itself. He just saw the data and he said, we need to look there. It ended up being right in the pocket. This is really cool. We already knew that, though. It would be really nice to have this information if you didn't know where your drug was binding. But that was a very high concentration of drug. what about lower concentrations? You know, we were basically asking a lot of these variants to knock off 10 micromolar inhibitor. So we did the experiment where we slowly walked back the concentration of drug, hoping that we might capture additional variant effects and drug resistance profiles. And so if this movie plays, you will see just that. So as the movie's playing, we're lowering the concentration of drug, and we're sort of expanding the shell of drug-resistant residues, and we're starting to capture second and third order drug resistance profiles, in some cases, positions that are nowhere even close to the contacts of the drug itself. So next we asked, okay, do we have the resolution to distinguish drug resistance and binding profiles of two very similar compounds? So we did that assay with the second inhibitor. And here's a scatter plot where the colored residues represent variants that are resistant to one, the other are both inhibitor in purple. So we capture all three classes. We can look at this in 3D as in before, get a good idea for the spatial distribution of how these align to these two compounds, both of which have been solved independently as structures. And one thing you'll notice here is that there's quite a bit of purple. So there's a lot of positions that confer drug resistance to both compounds. And while there's been a lot of talks here and a lot of interest in general about defining positional patterns and trends, we wanted to know under the hood what's happening, what variants at those positions are actually affecting one or both drugs. And so we generated what we're calling DMS chemical fingerprints, but basically a small heat map, just looking at the drug-resistant variants in this region, and asked, okay, now when we color by whether it affects inhibitor 1, 2, or both, what kind of patterns emerge? And so this is giving us a better chemical insight into the drug resistance profiles in this assay. And in positions like 671, for instance, where you see a number of variants that are resistant to both compounds, you also see drug-resistant variants that only hit inhibitor 1. That's a really good case study of something that was already known through years of medicinal chemistry and structural efforts. So we were really reaffirmed in that moment. But everything I've talked about right now has involved breaking drugs. And so for the last bit of this talk, I just want to touch really briefly on what we're most excited about, which is, can we do the reverse? Can we find variants that are actually increasing the potency or efficacy of drugs and use that to help guide chemical design? In order to do that, we tested lower concentrations of both of these inhibitors, and we capture less, but we do capture some. We, again, capture classes, all three classes of affecting one, the other, or both inhibitors. We can look at these again in 3D, get under the hood again, and look at how their chemical profiles behave relative to one another. And the last thing I'll say here that really got us jumping out of our seats is that we noticed in a few positions there were potentiating variant effects that were occurring at positions that were already important for the other compound. In other words, this position 739 on the right here is known to be critical for inhibitor 1 binding the red inhibitor, but not for the other. And at that position, there are two variants that appear to be potentially recreating a contact or basically recreating the existing interaction that inhibitor 1 had. And so it's exactly this kind of data that we're really excited about. If we can understand this correctly and start building models around this, then we can use this as vectors to guide more smartly guide our chemical optimization and searching. And so, yeah, in summary, I talked a bit about the allosteric site identification and figuring out therapeutic sites of action, and then getting a better understanding of protein-ligand interactions using DMS, ultimately with the goal of making better treatments for patients. And there's a ton of people involved in this project over the course of 2 years, both on the BMS and Octane side, so I want to thank all of them. Thank you for listening, and I'll take some questions.
Thanks, Conor. Brilliant. I think it's one of the first times I've seen any data like that. I don't know whether other people have seen something as systematic as that with drugs. I haven't. Okay, questions?
I've got a question over here. Yeah, and I have the microscope, or microphone. Not a microscope. Super cool talk, Conor. I'd love you to just think with you. How would you, because we're doing this with GPCRs right now, and then we're struggling to think how would you actually design a ligand based on this information? So would you add a single little not to get you in trouble, but get you in trouble?
That's a very good question. I don't know if you have insider information, but we are actively trying to do this. After we got these results, we were chomping at the bit. We are currently, we have compounds that we designed for GPCRs that are currently being synthesized to test exactly that. The short answer is it's really challenging. I can throw out AI, ML, and all these other buzzwords and say that we're going to wrap all of the information we can into a model. But at the end of the day, one of the most useful things was to engage some really smart computational chemists to look at the data and manually inspect the results and just think about each position, especially ones that are, very enticing as potentiators. And we did that for a chaperone, for a GPCR, and we have 10 hits that we're ordering, and we're really hoping basically close the loop and validate this model so that it can become yet another step in our iterative process of high throughput scanning.
Cool, thanks.
One more quick question.
Okay, hopefully this will be quick. Thanks for the really good talk. This is Grant from UCSF, Grant Goldman. I was really interested in the idea that we observe people walking around with mutations in TYK2, and I'm just wondering if your experiments recapitulated any of those protective effects and where those mutations fall in the protein.
Yeah, that's a very good question. I'll give a short and maybe a much longer answer later. It's hard to say whether we captured the protective effects, because part of what we were seeking to find out is what are these protective alleles linked to. So there's one in particular that's very prevalent, and we went into this project aiming to test multiple different cytokine pathways and figure out, okay, this is maybe it has some biased effect. It only affects one or some pathways and not others. And then we could build a genotype-phenotype relationship from there. In the end, we actually got the opposite. Every pathway we tested, it had a very minor effect on activity in a non-significant way. The only assay that it had a very clear effect on was the abundance or stability assay. So that's something that we're still actively working on, we're really excited about. But in building burden tests and other analyses through that data, we're starting to find out that some of the best phenotype-genotype correlations are actually more linked to TYK2 abundance than specific signaling activity. And there's lots we could wildly speculate on about whether in some tissue-specific instances, certain levels are more sensitive to activity or abundance than others, but yeah, work in progress.
Okay, thanks very much, Conor.
Thank you.